Hello, and welcome to Let's Talk SADS Live, Canada. I'm your host, Dr. Shu Sanitani, and delighted to be coming to you from Vancouver here in the morning, but wherever we are reaching you, watching this in the comfort of your home or sneaking it at work, welcome to our monthly webcast. We were delighted to add an extra session this month and had great questions asked by our audience and had a little Q&A. So very enjoyable. So this is a space where we come together to learn together collaboratively, learn from each other. I do a ton of learning here, and always great to talk to the guests. We've had some wonderful people come and donate their time to share expertise about sudden arrhythmia death syndromes, SADS conditions, which of course affect so many of our audience, either family members or patients themselves, healthcare providers. So we're really here just to talk about SADS conditions and bring you hopefully interesting insights, and also cover some of the basics, because there's so much misinformation out there everywhere, and information is readily available. Yesterday I was in the emergency department and telling the family about a new diagnosis, and they were literally on Google while I was talking to check my work. So we are here to give you the best information possible. And so we've got a special show for you today. Just a little comment about the SADS Foundation in Canada. So we've existed for 3 decades almost, through the dedication of Pam Husband, who's our executive director. And her mission really has been to serve our SADS population and allow a forum for networking, resource finding. And it's in the spirit of this that we have a website that's slowly growing some depth, and we have a clinical trials page. And so there's a study there which is called the Target trial, which is the subject of which is arrhythmogenic right ventricular cardiomyopathy, which is of course what we're going to spend a lot of time talking about today. So check out the website. There's lots of other cool things there as well, of course. You may be eligible, or you may be a healthcare provider and you want to stay on top of things, because our patients, of course, are coming to us and saying, what about this? Or I heard this. And I'll just take a moment to put a plug into our great friends south, the SADS Foundation in the US, with Alice Lara and Mike Ackerman and Aaron Waite, who all support us being here. And they're in the midst of another big organization to be presenting to the FDA about SADS conditions. So lots of activity going on. So as you know, I sort of like to add a little bit of homework for you and a bit of fun learning here. Sorry, my slide's a little messy there. It said, did you remember that but it's all messed up. But basically this time I'm telling you some facts about SADS conditions. So we talked about that catecholinergic polymorphic ventricular tachycardia can look like digoxin toxicity, so the up and down of the abnormal rhythm. Arrhythmogenic cardiomyopathy, of course, was described hundreds of years ago. Sir Francis d'Assisi, the Pope's physician, made a description of it in the 18th century. And then last time I reminded us that even though Brugada syndrome primarily affects adults and primarily males, the original report actually had 3 of the 8 patients were children under 10, which is of course uncommon in current day Brugada series, but not uncommon because children are often affected quite significantly by SADS conditions and may be the first to present in a family. And so today's fact is, did you know that arrhythmogenic right ventricular dysplasia, ARVD, which some of you may have been initially diagnosed with ARVD, and then it was reclassified in 1995 to a cardiomyopathy, or a heart muscle condition. So this is from a thoughtful document that said, well, actually, this is a heart muscle condition as opposed to a degenerative or a cancerous type transformation. So this is a heart muscle condition. So that's our fact about SADS. And on that note, arrhythmogenic right ventricular dysplasia, now known as arrhythmogenic cardiomyopathy. I'm delighted to have our guest today, Dr. Kathleen Hodgkinson, and she's already given me permission to call her Kathy. She is really known across Canada and outside of Canada as the foremost genetic researcher in arrhythmogenic cardiomyopathy in the genetic counseling background. And so she got her degree in genetics and cell biology at the University of Manchester, and then received Rotary and Commonwealth scholarships, and then graduated as a genetic counselor from McGill University. So lots of work here, but I'd rather bring her into the studio and get her to take us through a bit of this story herself. So please join me in welcoming Dr. Kathy Hodgkinson. Kathy, welcome.
Thank you very much, Stu. Thank you. We're at opposite sides of the country, are we not?
We are, and I'm sad to say I've never been to Newfoundland, but it's certainly a goal, and we of course communicate across the hours because you are such a wonderful resource. I didn't tell the audience that you're also on the medical advisory board for the Canadian SADS Foundation. So to get us started here, just give us a little bit of your journey. I always find it very interesting for our audience to understand who we're learning from today. So your education started in Manchester, and you were a genetic counselor well before it was on vogue to be a genetic counselor.
Sure, you're letting everybody know just how old I actually am. You're right, that's true. Yes, but and certainly we had no genetic counsellors in England, which I think was very helpful in getting both the scholarships that I got to come to McGill, which were an enormous help. And I think that people who offer scholarships to students, in this case Rotary, in this case the Commonwealth Foundation, are so wonderful in that they allow people to learn from in places when they don't have that particular learning opportunity from where they live currently. And that was certainly what happened to me in the UK. But I actually arrived in Newfoundland at the end of 1995, 1996, and it was because of the Canadian Genetic Diseases Network that we're recognizing that Newfoundland is a genetic isolate. It's also known as a founder population. And because of that, there'd already been lots of great work here by Dr. Jane Green, who had done a lot of work with Lynch syndrome and some of the hereditary cancers that, again, run through families in the same way that ARVC does, and DCM and HCM. And because of that, there was a recognition that there were diseases here in Newfoundland that we had not really understood. We didn't really have a reason, a genetic cause for them being, what was their cause. So I came here to have a look initially at the cardiac diseases and actually neurological diseases as well. But the cardiac diseases became very quickly the ones to focus on, and primarily because the outcome for not diagnosing a cardiac condition because of a genetically high risk sadly often resulted in death, and that is unacceptable. So that was one of the main reasons that when I first arrived in Newfoundland, I took on the cardiac conditions, because they were so their impact was so dreadful. And talking to families at that time, in the end of the 1990s, families were particularly scared to get phone calls late at night, because if they did, their fear was that somebody else had died, and that somebody else young had died, because they recognized that happened in their families. So that was sort of you're right, my background was Manchester and McGill, and I worked in Oxford for a short while. Then back to Toronto, and then came to Newfoundland in the end of the 1990s, and have been here ever since. I think I came for 2 years actually, but it's longer than 2 years.
Yes, and you've found a wonderful home. And so we've already sort of dipped our toe into what we're going to focus on today. So arrhythmogenic right ventricular cardiomyopathy, and so you must have there must have been in Newfoundland this sort of almost folklore of these young men, especially, who would tragically die really at the prime of their lives. And so how did you go about unraveling this? Because of course you are a huge part of how we now have so much greater understanding of what's actually going on.
I think, first of all, you're absolutely right. There is a folklore associated with it, and in some families it's called the curse of the whatever the family name is, and it's a curse. People would say things like, young men don't live long in our family, they die young, and they don't drown. Because another reason for young men dying in Newfoundland because of the fishery is sadly drownings, is sort of fishery related deaths. But these young men died and it was nothing to do with the fishery. So I started with a very small family tree, actually, that had been that was known in the provincial medical genetics program at the time, and basically built that tree up. So followed what we call, as a geneticist, I follow the disease line of risk. So I will follow the disease through the family. So I don't follow both parents and then both their parents. I follow the disease. And in that way, you can build up very quickly, actually, which did happen, a family that was over 10 generations and over 1,200 people as one family. And what that allows you to do is it allows you to see the disease running across generations. You can see, because they have family history, because these deaths occurred and they're so unusual, that family history is remembered. It's written down in family Bibles. It's on the gravestones of local churchyards. You can find these ancestors and you can see their date of birth and their date of death, and they died in their 20s or their 30s. So you can very easily build up this family tree with the help of the families who are with us now. And the families at the time, of course, were so keen to try and find an answer to what they knew was in their family, and which was not necessarily taken that seriously. And we can imagine that, can't we? We can imagine a young person in their early 20s who feels a little faint, a bit pre-syncopal, felt a bit wobbly, knows this is in the family, goes to the emergency room, all the clinical tests are normal, they look healthy, they run, they're told to go away because they're healthy, because they're okay. But actually, some of those individuals died because they weren't okay. They had the variant, the genetic mutation that we eventually found. And you can have normal cardiac tests and still be at high risk of the first event happening to you being death. And that was the impetus for all of us here to really try as hard as we could to find the cause of this dreadful disease.
And
I was going to say, I will fill you in with a story, and I know the family, I'm obviously not going to name them, but a tragedy occurred where 2 brothers died within 8 days of one another. One of the brothers brought the body of his brother home from another part of Canada, and at the meal before the funeral of the brother who died, sadly that brother died. Now it takes a lot to remember. It is unbelievable, and it's beyond tragic. It really is beyond tragic. How do you even sort of understand that sort of occurrence? And so those were the stories that to me meant this is ridiculous. There must be a way. There's a gene here clearly, and we knew that because geneticists, what do? We look at patterns in families, and the pattern in the family was clearly autosomal dominant, clearly running vertically across the generations, male to male transmission, which clearly tells you it's autosomal dominant. And for those people who remember their biology, men pass their Y chromosome onto their boys. So unless a gene is on the Y, and there aren't many genes on the Y chromosome, it's an autosomal dominant disorder. It's not X-linked. Because that would be the other reason, wouldn't it? If boys get affected more than girls, you might think it's X-linked. It's something to do with the X chromosome. And, you know, a bit like haemophilia, which affects boys but doesn't affect girls. But it isn't. It's dominant. It sits on a chromosome. It's a 1 in 2 risk from a parent to a child, and that's why you see this vertical transmission across families. So
let's just drill.
Sorry,
let's just drill down on that a little bit, because some of our audience isn't as genetically literate as others, and so that's such an important concept, because you know, one might think, you know, just on the stories that, well, clearly this is something that's just affecting the men, and so the women could carry it and only kind of give it to the men. But the fact then that you have, exactly as you described, you have men being able to pass it on to their sons, so it cannot be on the X chromosome. Some of the muscular dystrophies, for example, those are an example that comes to mind, or some of the other conditions where a female can carry it. But this is different. This is on the non-sex chromosomes, but still the men are much more significantly impacted.
They are, but once you pull together these very large families, and just to put this into context, we now have 27 families here. So one of the big ones is the one I described, which is very, very large, our largest family, but most families have a few hundred people in them. We do see women who have died in their 30s quite often. So it's not insignificant in women, it's just not as bad as the men. And the data we've already published quite clearly shows that if you look at women who have, we haven't according to our talking, we haven't found the gene yet, but we did find it. I'll just press on that. So when you know who has it and who doesn't, you can quite clearly see that the women who have this genetic variant die sooner than their unaffected sisters. The men who have this variant die sooner than their unaffected brothers, but they die a lot quicker than the women. There's a big sex divide, but the women still die quicker than their unaffected sisters. So it's not insignificant in women. It's just not as significant as it is in men.
Yes, thank you. And I think the signal is so disproportionately strong in men. I don't think there's any condition that is as as penetrant as the males with this, and we'll come back to that. The other thing I just want to touch on, because I'm always conscious of, I don't want to panic our audience, but this notion that one can have fairly advanced arrhythmogenic cardiomyopathy, but standard tests are normal. And that's where the family history is so important, because of course, if you have a family history, so if you have a parent who has a diagnosis of arrhythmogenic cardiomyopathy, you have a 50% chance of inheriting it. Even if your initial tests are normal, you still warrant follow-up, because this cardiomyopathy can present later in life, and the tests can be very nuanced.
What I really would like to emphasize as well, which I think has to be very clear, is that the disease we talked about and which we found here in Newfoundland is known as ARVD5. It still has the D because that's the way it was originally defined. In other words, the fifth one to be found. It is so different in many ways to any of the others. So it matters what gene is involved. So for all the audience, if you have a PKP2 variant or a DSP variant, and we're talking about ARVC now, or whatever variant in, you know, whether it's CPVT or long QT syndrome or HCM or DCM, the gene involved matters and the variant in that gene matters. So a gene, of course, is a message. It's a message that can be very long or very short, but it has a length. And you can have variants completely along that gene, very different variants. And some variants will be disease causing and some won't. Some will have no impact whatsoever. The TMEM43 gene and protein that was found to be a cause of this particular nasty sudden cardiac death disease in Newfoundland has one specific variant in it. It's a, doesn't really matter what it is, but it's a PS358L. So it's a change of one amino acid, a serine, to a leucine. So there's a bit of a change in the protein. And it's one little change from a C to a T. It's a single letter change in the DNA code. And that alone, which is so tiny, has this enormous effect. And we really don't see any other variants in that gene that have this same impact. Whether they turn up, because of course, we learn new things every day. People publish new things every day. So it will be interesting to see. But the way I'm describing this particular variant, this particular gene, pertains to this gene. It does not pertain to a PKP2 variant or a DSP variant. So you do have to know what type of disease you have. And I think that the field is moving towards a gene-based approach, isn't it? So rather than a morphology-based approach, which is you have ARVC, which I think will soon be renamed as arrhythmogenic cardiomyopathy. So it takes away the R, because the R, some people have an L, the left ventricle. So some people, you don't see any structural change in either. So the name, I think, will change. And we will start to look at these by variant. So it matters which variant you have as to what happens to you. So for this variant here in Newfoundland, if you are a man with this variant, you will get a defibrillator. You will be offered a defibrillator based on the genetic test alone. You don't have to have anything else. Your heart can be completely normal by every test, but you will be offered an ICD at the age of 18, and we have implanted them earlier than 18.
Let's just backtrack a little, because we're getting a little bit ahead of my understanding, and I want to savor this story a little bit, because of course you have this now multi-generation disease that you've tracked.
Yeah.
Take us back a little bit, because obviously when you so first you did linkage studies, and then you've identified possible candidates, and then people said, okay, TMEM, transmembrane protein 43. I won't even ask if there are actually 42 other ones, but so here's this TMEM 43. What was your reaction? Tell us more about that story when you found that this is a suspected genetic cause of this, and how does this fit in? Because it is a different type of ARVC or ACM from the other ones.
In many ways, it is. If you can imagine it, we sort of had started to look at this, as I say, from the mid 1990s. Dr. Sean Connors, who people, if they're listening here in Newfoundland, will obviously know him. He's head of cardiology here and has been responsible for looking after all our individuals here. He and I started working together in 1998, and it really was at the start of the 2000s that with linkage analysis, and this is not the forum to go into linkage analysis, but just to say that with linkage, which is a research tool, you can define people as being at very high risk of having the mutation, or at very low risk. But because you don't know, it's a bit like knowing a street address. You know that somebody lives on Circular Road, but you don't know the number of the house. But you can see that it's Circular Road versus Empire Avenue or Newfoundland Drive. Or so linkage allows you to be quite clear that you look like you've got Circular Road. So we think you've got this variant because the house should be on there. But because we don't have the house number, we're still a bit unsure. So we used linkage analysis from 2000 to when the gene was found we used that to implant ICDs in people. So we were very proactive in using actually a research result, which is highly unusual. And really the only way we could do that was because our team here is so tight, and Terry Young, who was the molecular geneticist who was in charge of the lab, we were absolutely clear that her and her, at the time, doctoral student knew exactly what they were doing. We could use that to implant ICDs. For people who it looked like they didn't have the street address, like they didn't have Circular Avenue, you didn't not follow them. You still followed them because we didn't have the mutation. We didn't actually have the actual gene. So we still followed them. Then I remember this very well. We were sitting in a small office, no windows, and we'd come down to in the previous weeks, the last 20 possible genes that could be the gene that caused this. And we literally had gone through 19 and it wasn't that. So we were at the 20th. And if that, well, we didn't know what we were going to do, but we were going through. Nancy, who was the PhD student, was reading out affected. In other words, this TMEM43 was abnormal. We had the variant there. She'd say, affected, and I would be looking at the pedigree because, of course, they're kept separate. You don't mix them. Nancy can't know who the person is, so she's not. Nobody is influenced in any way. So we went through them one by one, and there were hundreds of people. And as we kept going through the list, nothing was wrong. So if Nancy said unaffected, I would say unaffected. If Nancy said affected, I would say affected. So you can imagine, even now, I have a bit of a cold, feeling going down my neck because we were getting to the end of this list and nothing was wrong so far. We got to the end of the list. Everything tallied, and there was silence in the room. I think there were about 5 of us there. We were just, oh my goodness, we think we've got it. This is it. There were tears shed, actually, because over the years and the decade prior to that, we'd all heard of the stories. We'd all, been privy to the most dreadful things happen to families, and it mattered. This mattered. It meant more than, yes, we'd done very well with the research, and yes, wasn't this great, and it would be a great paper and all. None of that mattered. It was literally, oh my goodness, we think we've got it. And there was just silence. I'll never forget it. And that feeling of cold water dripping down your neck. Yeah, it was a eureka moment. It was. And yeah, I've never lived through anything like that previously, and I'm almost certainly never going to live through that again. But yeah.
Well, and thank you so much for taking us back. I think, I think it's a tremendous privilege for me and for our audience to hear, first of all, the incredibly hard work that goes in, because back in that era, the techniques for sequencing and for isolating DNA and stuff was not what it is today, where you have, now you can, it's a much more automated, mass scales. This was people in, with pipettes and measuring things, and, an incredible amount of work. And then, running gels all the time, and then amazing. And so, and, to our audience, Canadians have done an amazing amount of work in the SAD space, in my view, in my very biased view. But there aren't that many amongst us who say, we identified the gene that was associated with a very important SADS condition. And so we commend Kathy and the team. Of course, these are all team things, and we've heard a few of the names. And yes, what an amazing moment that would have been. And let's fast forward now, because you've said, identification of that gene. There's a care pathway, and that care pathway I want to come back to. But are we just screening known families, or is this so common that the family doctor is saying, I'm going to send you for the Newfoundland ARVD gene tests, or who's doing the testing?
That's an extremely good question. We actually have published a little bit on. We did a public perception study asking what would people think about a population screen, because you might argue that this is one of those things that you might screen the population for. So just do it doesn't matter what your family history is, we should just screen for it. We don't do that, by the way. This was just us asking the population what they felt, and the results from that were the population would be quite happy with that of the people we had spoken with. But no, it really is just families that we're looking at. Either it's cascade screening for within families, or quite often if new families arise at a new early sudden cardiac death, the first thing to be checked for is this. Or somebody coming in, we will check most of the DCMs actually as well for this, because we have had a few occasions where the presenting sort of clinical picture was DCM. Unusual, not common, but it has happened. So we will exclude this variant as a first pass in anybody that's really coming to the cardiac genetics clinic with a history of sudden cardiac death and or of DCM and or of unknown arrhythmias. We would automatically exclude it as a first pass.
And just for our audience, DCM, of course, dilated cardiomyopathy, which can sometimes mimic an arrhythmogenic cardiomyopathy. They definitely share some commonalities, and to my understanding, we don't fully understand. So the vast majority of ACM, arrhythmogenic cardiomyopathy, where we understand the genetics, most of it is in the structural, in what we call the desmosome. So what's sort of holding the cells, the heart cells together and allowing the communication. But to our knowledge so far, Team M43 is not associated with that form of arrhythmogenic cardiomyopathy. The other thing sorry, please carry on.
Actually, I was going to say, ironically, to say that we found this gene in 2007, 2008, we still don't know where TMEM43 actually sits in the cell. We still don't know. We know it's membrane bound, but we don't know which membrane it's on. We don't know whether it's the inner nuclear membrane, the outer nuclear membrane, the endoplasmic reticulum, the outer sort of cell membrane. We don't know. And so I think there's an awful lot about, I think this protein is important because of the effect that this particular variant has, but there's still some of the basic information we just don't understand. And what I think is nice, and I think sometimes being in a small place can have its downsides. Getting out of Newfoundland can be quite hard when the weather's bad. Getting back in can be very hard too. But you do pull a team together, and we are very close as a team because we're actually located next to one another. We're in the same place. And one of our new colleagues, Jessica Esseltine, who is a functional biologist, is doing induced pluripotent stem cell work with this population. So if we think of our TMEM43 population, we have men who are very badly affected early on. We have some men who don't get as badly affected. They can live, relatively normal lifespan. And we have women who get sort of again are the worst affected of their group, and women who are not very badly affected at all. And we have, as I said, their unaffected siblings. And so what has happened is we've been able to take biopsies, skin biopsies from these different patients. And Jessica has grown them in the laboratory and has grown fibroblasts, which is what you grow from skin cells, the skin cells themselves. And then she turns them back into stem cells. And stem cells, are those cells that could become any cell. They are pluripotent. They can be anything. And then pushes them forward to be cardiomyocytes. So what you can then have in the dish is you can have your cells from somebody who has a very bad disease process, somebody who doesn't, matched for sex, matched for their siblings, and we can see the differences in the cells in the dish. And in the dish, you can then CRISPR, the gene editing mechanism. We can CRISPR the variant in or out. So we can take it out of the person who has it. We can add it to their brother or sister who doesn't have it and see what happens there. So I think that what has really given me great joy really is that this project is not over. This whole our learning is nowhere near done, and we have junior faculty who are able to sort of pick up the torch and run to sort of learn as much as we can to take us to those next levels as to what is this actually doing in a cardiac cell, which you're right, at the moment, we still really have no clue at all.
Yeah, no, and I'll take a minute to plug we had a speaker last year, Dr. Glenn Tibbetts, who really took us through the stem cell process and science. And so, I'll refer our audience back to the archives, and they can pull out that episode. And really, I mean, the science, and again, fixing the DNA problem or introducing it to study exactly what these seemingly very localized, very simple amino acid changes, and they impact an entire civilization, as it were, in your part of Canada. Now, we talked about, you mentioned, if you have this gene, we'll typically offer an ICD, an implantable cardioverter defibrillator, that can deliver therapies. And one of the things, of course, that SADS Canada is anxious to support and eager to support, of course, are these much more sophisticated therapies. I think, a defibrillator is sort of like the fire extinguisher. Once the thing is burning, we need something to put it out. But, how do we prevent the fire from even starting?
Yeah.
And that's where we get into the stem cell work and research, really looking at the mechanism. In the young people with these defibrillators, what are some of the learnings there? Presumably they're getting shocks from their defibrillators, but I imagine it's been life-saving as in most of the people.
Well, it has, and I think you're absolutely right. An ICD is a fire extinguisher, and you really would like not to ever have to use a fire extinguisher, wouldn't you? I mean, it just means there's a fire, and nobody wants a fire. So work towards actually stopping the actual disease process, so you don't actually need the ICD, is really where we need to be going, because the ICD. And I'm sure many people you've had on your show before will have talked about. Obviously, the ICD can fire for inappropriate reasons. It's nothing to do with the disease process. Having a shock from an ICD is not value neutral. It can be very devastating. We have actually looked at our population here and asked the question of spouses of people with ICDs, the people with ICDs themselves, and their negative siblings. And all of them score they score clearly higher than population levels for depression, anxiety, and PTSD. And you'd sort of expect that, because the psychological burden of the ICD is quite significant, I think, and particularly for young people. So anything we can do, and I think, the target study that Jason Roberts is running, again, you want to see these studies occur where we might be doing something that actually alters the way that disease is progressing. So the ICD is needed a lot less. I mean, ultimately, you'd like to cure it, wouldn't you? But, probably a long way down the line. But that you would like to see. And so I think you're right. We've been very blessed in Canada to have some great people around who do, your guys, and there's you out in BC, and Andrew Cron, and Laura Arbour, who's done some phenomenal work out there, and across the country, really good people. And I would just like to say here that, I met Pam originally in the late 1990s, and we neither of us look any different as when we first met, and I'm sure she'll agree with that. But Canada's lucky to have Pam Husband. It really is. She has done this job based on a tragedy in her own family. Really, there aren't enough accolades really for the Pam Husbands of this world.
Oh, well, thank you, Kathy. And I'm sure Pam is backstage in her very modest manner saying, not at all. But I completely agree. And I met Pam around that era. So I completely agree. In our last couple of minutes, I just want to scratch the surface of something without necessarily going down fully in the rabbit hole. But I read recently that, exercise restriction is a big, it's sort of it's something that's coming down from on high, that, if you have a genetic risk for arrhythmogenic cardiomyopathy, you should limit your exercise, because exercise will accelerate your disease. Now, of course, there's lots of benefits to exercise. How's the team handling that? Someone, might be diagnosed when they're, 5 years old or 10 years old. Are you having success? Are you having conversations where you say, thou shalt do, in the words of Mike Ackerman, thou shalt do, cricket and rifflery and that? Or
cricket is quite as I'm a fan of cricket, I think you do raise your heart rate at cricket, but it's one of my favorite games, actually. So I do like cricket, but it's a very good question, and I'm really not the right person to answer it, really, because our pediatric cardiologists, Heather and Megan, would be much better at answering this. But my understanding is we have looked at exercise in this population, and we have shown a relationship between exercise and the way we did it was looking at firings of the ICD, and people who exercise a lot get far more firings, appropriate firings of their ICD. They're not inappropriate, they're appropriate. So it looks like exercise is having a bad effect. And we've only changed our policy relatively recently to be testing children under the age of 5. And the reason for that was because of the psychological sequelae that occur if you are having to tell a 15-year-old that their dream of being in the NHL or whatever sort of elite sport they want to be in is now not going to happen because they're now being exercise restricted. So I think the moving of the testing to a much younger age is potentially going to hopefully ameliorate some of that. And I know some of the families have said that what they're trying to do with their children who they want to do exercise and they want to get them out there is they've put them in golf and curling, actually, because again, they're very competitive. And of course, Newfoundland, Brad Gushu's team has just done extremely well and won his 6th Brier. Well done. It's great. I mean, you can do very well at a sport which is very difficult, very competitive, but just does not increase the heart rate to the same extent as in the NHL or basketball or volleyball or soccer. So I think parents seem to be taking that in that vein, but I think it's too young, really. And as I say, I am not the person who has the most experience with that.
Well, on that note, I'm afraid we're at time, but a great reason to have ongoing discussions. And, just to circle back, we don't actually understand what the protein does, but somehow it is behaving similarly to the ones where we can sort of conceptually, we can understand if if the structure of the cells is, how cells are held together is affected by exercise. How does
Then
how
you
does
can
this
understand
protein?
it. Yeah,
Yeah.
yeah, yeah.
Kathy,
I
thank you
would
so
like
much.
to actually,
Oh,
could I
yes.
just, I just would like to say that to thank every family and every person who ever participates in research. Nothing would happen without you at all. And it's really, I've been so blessed by meeting the most amazing people over this last 25 years. So I just wanted to make sure that I'd said thank you.
Oh, well, we're happy to pass that on. It's such an important point, and families probably wonder why we're coming at them with stacks and stacks of. We're still mostly on paper here and that, but it is the only way to learn and to advance the science, and it is the researchers. So, lobby for funding good SADS research. So, Kathy, thank you
Yeah.
so much, not
Thank you.
just for being on the show today, but really for all that you've done. And it is such an honor to have you talk and share some of this story. It's an amazing story, and I want to I can hear more and more details about it. It's just fascinating. I will get out to that part of the world to meet some of
Please
these
do. You're
families.
welcome anytime. You know that, and we'll take you whale watching, and we'll screech you in. So.
Fantastic. All right. Well, thank you. And what an interesting show, and such a Canadian story. They have found the same variant in populations outside of Newfoundland, but not nearly studied to the extent and not nearly as numerous. So um, it really lots to learn still ahead. So thanks for joining. I know we have another great episode ahead for you in April, and I do hope you will continue to consider supporting the Canadian SADS Foundation to allow us to keep broadcasting and support collaborative learning. We're still figuring out how to get together in person, and thanks for your attention today.